Hello and welcome to this week's lesson in biology. We're working on genetics, and this week in chapter 11 we'll be looking at section 3, chromosomes and human heredity, and chapter 13, applied genetics. I suggest you do the following while watching this video. Follow along in the book. The chapter and section numbers are at the top of the slides. Pause the video when necessary to find your place in the book, to listen again to something you didn't understand, or to work on one of the example problems. So our first subject is chromosomes and human heredity. A karyotype. It's a micrograph, or in other words, it's a picture taken with a microscope, in which the pairs of homologous chromosomes are arranged in decreasing size. Images of chromosomes are stained during metaphase. In metaphase, the chromosomes have swelled up and they're big and they're thick and they're easy to see in the cell, and a stain, a dye, is used to make them visible. And the chromosomes are then arranged in decreasing size to produce this micrograph or picture. Telomeres. Telomere caps consist of DNA associated with proteins. They serve as a protective function for the structure of the chromosome. Nondisjunction. This is cell division during which sister chromatids fail to separate properly, and they're responsible for Down syndrome, also called trisomy 21. Now, our other subject this week is applied genetics. And applied genetics is something that people have been doing for hundreds and thousands of years. The first type of applied genetics is called selective breeding. And it's the process by which desired traits of certain plants and animals are selected and passed on to their future generations. Hybridization. Hybrid organisms can be bred to be more disease resistant, to produce more offspring, and to grow faster. A disadvantage of hybridization is that it's time consuming and expensive. In hybridization, organisms with different traits are selected in order to try to produce an organism that combines both the desired traits. In general, hybrids are more disease resistant, healthier, and they do produce more offspring and grow faster. Inbreeding. This is a process in which two closely related organisms are bred to have desired traits and to eliminate the undesired ones in future generations. Purebreds are maintained by inbreeding. Now, there's a major disadvantage to inbreeding because it's harmful. It's harmful when recessive traits are passed on to future generations. Because of inbreeding, it makes it more likely that that an organism would get two recessive traits if that type of disease works in a family history. A test cross. This involves breeding an organism that has an unknown genotype with one that is homozygous recessive for the desired trait. And the reason for this is usually with plants, and it helps scientists figure out what genotype that unknown organism actually is, so that in future generations when it's bred they'll be able to predict what kind of individuals will be produced from that breeding. Okay, some formative questions here. What does a karyotype show? Is it A, the blood type of an individual, B, the locations of genes on a chromosome, C, the cell's chromosomes arranged in order, or D, the phenotype of individuals in a pedigree? If you said C, the cell's chromosomes arranged in order, you are correct. What is occurring in this diagram? Is it multiple alleles, non-disjunction, non-synapsis, or trisomy? And the answer is B, non-disjunction. What condition occurs when a person's cells have an extra copy of chromosome 21? Is it A, Down syndrome, B, Klinefelter syndrome, C, Tay-Sachs syndrome, or D, Turner syndrome? And the answer is A, Down syndrome. Which statement is not true of hybridization? A, it is relatively inexpensive to perform. B, it produces offspring with specific traits. C, it crosses a parent organism with the different forms of a trait. Or D, it can take a long time to be successful. The answer is A, it is relatively inexpensive to perform. Which term explains how humans have been able to produce a wide variety of domestic cats? Is it A, homogenization, B, inbreeding, C, selective breeding, or D, test crossing? The answer is C, selective breeding. A new breed of cattle has been developed by crossing English shorthorn cattle, which provide good beef but cannot withstand hot environments, and Brahman cattle from India that have a high heat tolerance but produce poor beef. The new breed, Santa Gertrudis, produces excellent beef and can live in hot environments. Which term describes Santa Gertrudis cattle? Is it A, crossbreed, B, hybrid, C, outbred, or D, purebred? The answer is B, hybrid. Harmful recessive traits can be passed through generations of purebred animals as a result of A, hybridization, B, inbreeding, C, line breeding, or D, outcrossing. B, inbreeding is the correct answer.